Okay, so let's also talk about acidity. This is another important chemical characteristic that we have to have some kind of handle on. Water, is it an acid or is it a base? Well, it can be both, and in fact is both. Water, we say that water ionizes. And here's a chemical equation. And again, let me stop here and say that I'm going to be using a lot of terminology and showing you a lot of structures that are unfamiliar to you. I really want to encourage you to relax, go with the flow. We'll fill in the blanks as we need to. I'm going to give you all the background that's required to understand this stuff, but don't feel like you have to understand the chemistry at a terribly high level. It's just not that important for our purposes today and for the class. But in any case, here's a chemical equation. Two water molecules form two compounds, two products. This is a hydronium ion and a hydroxide ion. And what we did was break off a proton from one molecule of water and attach it to this other water molecule, giving it a positive charge, and this hydroxyl is negatively charged. So what we see, when we say water ionizes, what we mean is that it really breaks apart. We can simplify that equation. If we look here, we see water breaks apart. These two arrows mean that it goes back and forth, or is said to be in equilibrium, breaks apart and forms a positively charged ion and a negatively charged ion. When we think about acidity, what we're talking about is an increase in the number of these protons, right? These positively charged hydronium ions. All right, now why is water pH 7? Well, we've probably all heard that pH 7, pH balanced scale. Well, what happens with water is that it ionizes just a little bit and it forms, again, these hydronium and hydroxide ions in equal concentration, right? Anytime you break up a water molecule, you got to form one of these and one of these. It just so happens that the extent to which water does that generates this number, 1 times 10 to the minus 7th moles or molar of hydroxide and hydronium ions. I'll refer you back to your text to think a little bit about concentration, but this is just a number that tells us how many of this either species is present in a given volume of water. And this exponent -7, that's a very small number. What that tells us is that water only ionizes to a very limited extent, right? But it does so and generates a particular pH. So here's a definition of pH. pH is defined as the negative log of the hydronium ion concentration. Well, if this is the hydronium ion concentration and we take the negative log of that number, what we get is a pH of 7. So that's why water is at pH 7 most of the time. Leaving water behind for a second, let's just think about what happens to hydronium ion and hydroxide ion concentration as we change pH, right? So if neutral is pH 7, neutral, now we know what that really means is that we have this concentration of a particular number of a particular species. If we increase the concentration, this line of hydronium ion, then we go down in pH, right? The negative log gets larger, the concentration gets larger and larger and larger. And we see here, this is again a logarithmic scale, again in terms of this molar units of concentration. Very small numbers of hydronium ions mean very high pHs, very basic solutions. High concentration of hydronium ions means low pH, very acidic. Okay. Conversely, the concentration of this hydroxide ion, as that increases, we see an increase in the pH into the basic range. So acidity, in one definition, is to extent to which a molecule ionizes and breaks apart into hydronium ion or hydroxide ion. So let's take a look at that. Yes, here's our definition. So acids dissociate, increasing the hydronium ion concentration. Bases dissociate, increasing the hydroxide ion concentration. So we said that water only ionizes weakly. With that number, the concentration of hydronium ions is relatively low, 1 times 10 to the minus 7 molar. For strong acids like hydrochloric acid, that dissociates completely into chloride ion and a hydronium ion. Whereas weak acids, acetic acid or vinegar, CH3COOH, dissociates only partially, like water, into hydronium ion or and the conjugate base, so-called acetate ion. In this case, we dissociate slightly more than water dissociates, but it's still very weak. And because acetic acid or vinegar only dissociates in a limited way, we would consider acetic acid to be a pretty weak acid, which is why it's perfectly safe to use in a kitchen context. Now, a word about chemical structures since we're here. Um, we're not going to talk so much about how we write these words down, these chemical words down, but this is a symbol for acetic acid, and the acid part is right here, COOH. That tells us that this is an acid group, and altogether though, this methyl, the CH3, we'll call that a methyl. Altogether, that's acetic acid. Okay, pHs of common household items. So we've talked about definitions of pH. We know that when molecules dissociate and release hydronium ions, the pH goes down, 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 becomes more and more acidic. Here's just a list of common items that you might encounter and the pHs of those solutions. So battery acid, very acidic, right? Very low pH. Lemon juice, fairly low pH. I don't see vinegar here, slightly higher pH. Orange juice also acidic. Beer acidic. Coffee acidic. Freshly distilled water, again, there's our anchor, neutral pH, pH 7. And then we get into the basic range, baking soda, slightly basic, right? We probably know that you can react vinegar and baking soda and get a vigorous effervescence of CO2. That's the base and the acid interacting with each other. Household ammonia, now we're starting to get very basic. Sodium hypochlorite, bleach, also very basic. But anyway, this gives you the sense of the pH range of some of the compounds that you might encounter in the kitchen, as well as some of the food items that we might encounter. Right, here's another list. Some weak acids that we'll commonly encounter in foods. And again, remember that solutions of these compounds are always going to give us a pH that's in the acid range, lower than 7. Carbonic acid, just dissolved CO2. Acetic acid, vinegar, butyric acid, something that you might be familiar with if you have rancid butter in your kitchen. Lactic acid, fermented lactose. We'll talk about this when we talk about fermentation. Citric acid, another one that we're common with. Tartaric acid, that's going to become important when we talk about fermentation of grapes to make wine. And on and on and on. Many different common acids. And again, what I want you to remember is that when we take these and dissolve them in water, they ionize and release hydronium ions into solution. Okay, so we've talked about physical properties, we've talked about covalent bonds between atoms, we've talked about non-covalent bonds that turn out to be very important. Let's talk about proteins, and we'll start our discussion of all of the biological molecules that we'll encounter with protein. And as I mentioned when we discussed eggs, the whites of which are mostly proteins become very, very important, and we encounter them again and again in all kinds of food preparations. So we want to start out with a description of what they are before we talk about what they do. So this requires us to talk about a little molecular biology. So let me direct your attention to the lower left-hand corner of the screen. This is the so-called central dogma of molecular biology. Our genes are encoded in DNA, deoxyribonucleic acid. That gets converted to RNA. But what the DNA really is encoding in those genes are protein sequences. So we have a whole set of biological processes that read that DNA and convert it into protein. So genes are pieces of DNA grouped together in three-letter codons, three-letter words called codons. And when we string those words together into a sentence, we get the sequence for a full protein. So proteins are polymers read from the DNA. What's a polymer? Well, a polymer is a string of monomers, right? Single units that are tacked together and perhaps repeating identical subunits, perhaps varying subunits. Proteins and DNA are both polymers. Now, going back to the DNA sequence, ATG, you guys are probably familiar now from watching enough CSI that ATGC, that's the sequence of DNA. This codon, this three-letter word, ATG, encodes for an amino acid called methionine, or we can write that as M. CTG encoding a leucine, or L, a different amino acid. CCC encoding proline, yet a third amino acid. So you can see that by stringing these codons together, these three-letter words together, we can build up a sequence of protein. Okay, now what's the protein look like? Okay, this might be our first actual complex molecular structure, and again, please don't let this intimidate you. I told you that biological molecules are made up of mostly carbon, you got some oxygen, you got some hydrogen. What's this R? Well, first of all, back up. Polymers are these long repeating strings of atoms and molecules. In protein, what we see is this repeating unit, and let's break it here at this double dashed line. There's one amino acid, and we form this thing called a peptide bond between the two amino acids. And now we have another element, right? Another amino acid linked together to the third with another peptide bond. So 3 amino acids in this polymer: methionine, leucine, proline. What's the difference? These all look exactly the same. Well, what's the element R? Well, this isn't actually a symbol for an element. This is called an R group, and it's representing the fact that at each of these positions we have a different chemical structure that distinguishes methionine from leucine from proline. And I'll show you those in just a minute, but just to remind you that what we're doing when we're making a protein out of a piece of DNA is stringing together amino acids in a polymer linked by peptide bonds, and each amino acid in that chain can have a different identity. Okay, that's a lot of molecular biology in 2 minutes. Is your head spinning yet? Don't worry. Let's look closer at amino acids before we string them together into polymers. So an amino acid is a structure that looks like this. This central point or vertex, there's actually a carbon atom there. It's sort of implied by this intersection of bonds. Each of these black bars is a bond. So the carbon atom is bound to this COOH, carboxylic or acid group. It's bound to this NH2, this amino group, and it's bound to this proton. And then the fourth atom can be another proton or hydrogen atom, or it can also be this R group or this side chain, the thing that gives the amino acid its identity. And then you remember that the peptide bond involves linking this amino group to the carboxylic acid group of the previous amino acid. So that's how we form our chain. So that's the building block of the protein polymer. Okay, you are probably also familiar from studying your cereal boxes that there are 20 common amino acid side chains, 20 different kinds of amino acids that we link together to form these complicated molecules called proteins. I'm categorizing them here for you. Let's skip that for just a second to look down at the bottom, just to give you a sense of what some of those different side chains might look like. So here in the bottom left we see the amino acid phenylalanine. Now that might be one that you've heard of before if you know about aspartame and the disease phenylketonuria. This amino acid is called tyrosine. It's just like a phenylalanine with this hydroxyl group tacked on the end. But let's take a closer look at phenylalanine again. Here's a ring structure, right? These two lines mean that they're double bonds between carbon atoms. Really, this is referred to as an aromatic structure. The electrons represented by those bonds are shared and distributed between all of the 6 atoms in this ring. This is also you might have know this as a benzene ring. So phenylalanine, benzene ring, side chain. You can remember the rest of that stuff dangling off at the end. Amino group, carboxylic acid group, alpha proton. Okay. Tyrosine, same thing, we've just added this hydroxyl. Here's a carboxylic acid group. You can see here that this molecule is ready to release its proton. Remember, the acidic functional group, we'll call it, ready to release that proton, increasing the hydronium ion concentration and making the solution more acidic. Here's yet another structure. This is lysine. And remember that each of these vertices represents a carbon atom. And actually, in this case, there's a carbon atom and 2 protons attached that are implied by this structure. But the interesting thing about this side chain is this amino group at the end, which can accept a proton, thereby decreasing the hydronium ion concentration and increasing the pH. But the point being that we've got a whole slew of different structures or different chemistries that we can form using different amino acids. But let's go back to this categorization that I've done. I'm showing you 4 categories of amino acid side chain. There's non-polar side chains. Those are, let's consider those to be oily or greasy side chains. There are polar side chains, those that like to interact with an aqueous or watery environment. And then a subset of polar side chains are acidic side chains and basic side chains. Excuse me. Now, the biggest distinction that I want to make for our class purposes is to say that this set of the 20 amino acids are greasy or oily. They interact not with water, but with each other or with oily, greasy environments, whereas all of these others like to interact with water. That tells you something important about biological structure in general. There seems to be this big dividing line between nonpolar functional groups, nonpolar chemistry, and polar chemistry. Let's take a closer look at that. But let's remember that each of these 20 amino acids has a different side chain. Okay, before we move on, here's a complicated slide. On the left, we see sequences of single letter amino acid codes, right? So this entire paragraph, if you will, is a single sequence for a single protein. That protein happens to be called GFP. There's a molecular structure of this protein. As you can imagine, this is much bigger and much more complicated to try to draw than, say, a water molecule. But what we can see is that there's serine, lysine, glycine. Each of these letters here is one of those 20 in the protein alphabet that we can draw on. The idea is that this sequence of letters differs from this sequence of letters. And what that means is that we get, from each individual unique sequence, we get a unique structure as well. So the sequence determines the structure. What's really important to note here is that proteins do things, right? What I didn't say when we were talking about the central dogma of molecular biology before is that proteins are actually the machinery of the organism, the cell. Though they carry out pretty much all of the work that the organism does at the molecular cellular level. The DNA is the information storage system, RNA is the information retrieval system. The proteins that are encoded in the DNA are the workhorse of the living system. And the way that we get different jobs done then is to use different genes that encode different protein sequences and therefore different structures. We won't talk so much right now about these structures. I don't think that comes into play for our purposes. But you can see that the structures are very different and very complicated. Oh yeah, they don't encode for one another. So this sequence will never form this other structure. The sequence entirely encodes its own unique structure and therefore function or job. I don't know if you guys can hear picking up in the background the thunder. Man, there's a huge thunderstorm rolling through today while I'm recording. Hopefully we don't lose power. Okay, onward. Okay, so while we're talking about proteins, let's talk about another non-covalent effect or interaction that becomes very, very important as we talk about food chemistry, and that is the hydrophobic effect. So what is hydrophobic? Well, look at that word and try to break it apart. So hydro, water, phobic, phobia, fear. So the hydrophobic effect is the effect of non-polar or water-hating or water-fearing chemical groups avoiding interaction with water. Let's back up 2 slides to remind you that the big distinction between amino acids is whether the side chains are non-polar or polar. What happens in proteins is that the hydrophobic effect makes sure that all of those hydrophobic or non-polar side chains in the protein get buried, hidden away from the structure, from the watery environment. So here on the left is another complicated picture of a protein. Every atom in the protein here is represented as a little ball. Not much sense to make out of that, except to say that I've colored all of the amino acids with polar side chains red or pink. And you can see that they're all exposed on the surface of the molecule and not much blue showing there. What I've done is draw all the nonpolar side chains, like phenylalanine, remember that benzene side chain, I've colored those blue. Now, what we can do is, with our handy-dandy molecular graphics programs, is slice that protein right down the middle as if it were an onion, and that's what I've done on the right. I've chopped away half of the molecule, and what you can see is that hydrophobic effect forces all of the nonpolar oily surface areas on the protein sequence to get buried inside, hidden away from water. I say that's the driving force in biological structure, and it's also going to be a very important driving force in cooking. I'll ask you to back up to cooking that egg again. Remember, egg white, clear, yellowish, proteins are all intact and dissolved. But when we change the environment, then we change the hydrophobic effect, basically, decreasing that effect and causing the exposure of those oily surface areas to the solution. Well, what happens then? They start glomming onto everything in sight. So you lose this unique biologically relevant functional structure in favor of non-specific sort of aggregation or glop, and that's what egg white is. So we lose this organization and we in favor of aggregates of proteins. So the hydrophobic effect is important for biological structure and for cooking.